As you go through the step-by-step approach in solving this paper to get maximum mark. The paper usually starts with an aim, or the question starts with an aim, and the aim of the question is always tied to the conclusion that will be drawn from it. To the conclusion. Here it says the investigation is looking at the effect of glucose concentration on the rate of anaerobic respiration in yeast. We know that when yeast respires anaerobically, glucose is broken down to give ethanol and carbon dioxide. And we can test this using a blue dye, which is methylene blue. It turns colorless with increasing concentration of carbon dioxide. So the time taken for the blue dye, the blue color to disappear in the methylene blue, can be used as a measure of the rate of respiration in yeast. Now the student's process involves, the first step is that there are 3 different concentrations of glucose labeled test tube 0.0, test tube 0.5, and test tube 1.0. So from here we can say that glucose here, since it's the effect of glucose concentration, glucose here acts or is the independent variable in this experiment. And they said the time taken to turn colorless can be measured. So the time taken will most probably be our dependent variable. So let's go through the process or the steps approach. We said put 5 cm cube of water in a test tube labeled 0.0, 2.5 cm cube of 1% glucose, and 2.5 cm cube of water to make 0.5, then 5 cm cube of 1% glucose to give you 1.0. That's how we made our 3 different concentrations. Now it says stir the content of the beaker containing yeast suspension with a glass rod. Add 5 cm cube. So the volume of it added was 5 cm cube to each of the test tubes. Put all test tubes into a water bath regulated at 40 degrees. So we can see some things that are being kept the same: the volume of yeast suspension, the temperature, the time. Also they said start a stop clock and wait for 3 minutes. So these are some values that we have kept the same in the experiment. After 3 minutes, remove the test tubes from the water bath and place them in a test tube rack. Use a pipette to add 1 drop of methylene blue. 1 drop was added of the blue dye to each of the test tube, and you stir with a glass rod. Use a second pipette to slowly add a layer of oil on the test tube. So we also add a layer of oil, 1 drop of test tube. These are the things that were kept the same throughout the experiment, and that's the picture we see here now, that we have 1 cm cube of the oil film, 1 cm cube of the oil film or layer of oil to maintain anaerobic conditions. So let's now go to the other part of the procedure. They said put the test tubes back into the water bath and restart the stop clock. Measure the time taken for the blue color in each of the test tube to disappear. So this is what we measure as our dependent variable. Alright, and record the time taken. The student stopped timing if the blue color had disappeared after 10 seconds. The recorded result as above 600. So that's our table of the result. Now the first question says prepare a table to record your result, the result of the investigation. Convert the time on the stop clock shown in figure 1.2 to seconds and record the time. So we are converting our time to seconds. And when we make tables, like I say, your table should start with a heading. You have headings, and with appropriate unit, you close all borders, and also you ensure that you use appropriate significant figures or decimal places. Alright, so let's make this table. Since we have to record only the test tube and the time, we need only 2 columns. So this we'll do use a ruler to do this. All right, we have our heading. Then of course we will insert our values. So here we have three test tubes. So test tube label. We have 0.0% test tube, 0.5%, and 1.0%. Of course, you close all borders like that. Ensure that you use a ruler to do this. And here we have time in seconds. So for the first one, they said we recorded it after 10 minutes, it's recorded as above 600. Of course, 10 minutes is 2 seconds will be 10 times 60, that will be 600 seconds. So we have above 600. Then the other one is 6 minutes 2 seconds. So we have 6 times 60 plus 2. 6 times 60 is 360 plus 2. So we have 362. So we have 362. And the other one is 2 minutes and 12 seconds. So we have 2 times 60. That's 120 plus 12, 132. So here we have 132. With this, our table is ready and we have our 4 marks. Remember, units are always indicated along with the table heading. All right, and that's the way to record your table. So you have your 4 marks. Here we'll go to the other questions. State a conclusion for the result of this investigation. Like I said, the conclusion must be tied to the aim of the investigation. We see that from the result, as the concentration of glucose increases, the rate of anaerobic respiration occurred faster. So we come here and we say higher glucose concentrations, higher glucose concentration higher glucose concentration increases, okay, the rate of anaerobic respiration, okay, and the yeast. All right, or use anywhere. Increasing glucose concentration increases rate of respiration, just to make sure that you don't lose the meaning of the conclusion. Now it says state the independent variable in this investigation. The independent variable here, of course, is the glucose concentration, because we changed it in and used 3 different concentrations: 0.0, 0.5, and 1.0. One variable kept the same. We saw that we noted a few variables that were kept the same. The temperature of the water bath was left at 40 degrees. The volume of yeast suspension added was 5 cm cube. 1 drop of methylene blue was used in each concentrations, and the layer of oil too was left at the same level. So these were the things we kept the same. So let's put one of those down. And it says, so we can say volume of yeast suspension used. Excuse me more. Volume of yeast suspension. All right, the other one says explain why it is important to study yeast suspension in step 5. It is for homogeneity, okay, to ensure, okay, equal number of yeast in each measured volume. So ensure that the yeast is evenly distributed in the 5 cm cube volume used. One way to improve this investigation would be to use an increased number of different concentrations of glucose. State 2 other ways to improve this investigation. We can ensure that we repeat the investigation for each level, repeat at each concentration. Okay, we can repeat the experiment at done twice or 3 times, and we take average. We ensure that we can also use a clean glass rod, clean or different glass rod, glass rod at each time, okay, each time, okay, in each test tube, so that the remaining liquid on each glass rod will not affect the result. So there are other things we can also keep the same. Now it says other things that we can actually do to improve the investigation. It says describe how you would make 5 cm cube of 0.25% glucose solution using 0.50 glucose. So the original glucose solution we have is 0.50. We want to reduce the concentration in percentage by half and make 5 cm cube of it. So we have to dilute it with water. So all you just do is you say take 2.5 cm cube of 0.25% glucose solution and add 2.5 cm cube of what? Distilled water. Okay, with this you make 5 cm cube 0.25% glucose solution. So half of the original 0.50 glucose, okay, will be made with half of the 0.5 and distilled water. So you can look at the video again to understand what we've done there. Now it says describe the method you use to test a solution for the presence of glucose. Glucose is a reducing sugar, and reducing sugar we use Benedict reagent. Benedict's, okay, reagent, and of course we apply heat. Benedict reagent plus heat. So color change, of course, will be seen. Color change from blue to orange or red, okay. But the important thing is that we use Benedict solution and we apply heat. Now it says student investigated the effect of temperature on the rate of respiration in yeast at 25 degrees and 35 degrees centigrade. It said in one experiment, the student measured the volume of carbon dioxide produced by the yeast every 5 minutes for a total of 30 minutes. Describe the suitable apparatus that could be used to collect and measure the volume of carbon dioxide produced.
This is what we do in the masterclass, really. We try as much as possible to ensure that we're able to properly, right, teach learners how they can properly understand, interpret the question as much as possible. We dive deeper than just a past paper to ensure that at the end of the day, they're able to fully understand any question that is being administered to them.
Apparatus to collect volume of gases or gases are many. We can use a gas syringe. That's a very easy and often used apparatus to collect the volume of gas. You can also collect in a delivery tube over water, okay, using a measuring cylinder. All right, you can collect it over water in a measuring cylinder from a delivery tube. But gas syringe is usually the easiest and simplest way, and that's 1 mark. So 1 point is just enough. Now let's go to next part of the question. So the student did there's 3 experiments at each temperature. They use the result to calculate the mean volume of carbon dioxide produced. Part of the student's results table for the experiment at 35 degrees centigrade is shown in the table 1.1, and that's the result. Now they said the student decided that the result of one of the experiments shown was anomalous. State what is meant by an anomalous result. When we say result is anomalous, it means it doesn't, a result that doesn't conform, okay, to the pattern, doesn't conform to the pattern of other results, okay, that is odd, okay, doesn't conform to the pattern. That's just a result that appears odd or different from the original pattern. And if you look at the results here, we have 1.8, 2.0. The one that appears not to conform here is 3.2. The difference is way more than what is for experiment 1 and experiment 3. Now it says describe how the students calculated the mean volume of carbon dioxide produced shown in the table. So when we do experiment twice or three times, we do that to exclude anomalous result. Since experiment 2 is anomalous, it's of course not included in the calculation. So we have 1.8 plus 2.0 to give 1.9. So we have 1.8 plus 2.0 divided by 2, and of course that's how the students calculated the mean. So we say we use experiment 1 plus experiment 2 all over 2. That is, we have 1.8 plus 1.8 plus 2.0, okay, all over 2 to give you the answer, which is 1.9, to give 1.9. So you can put this in words. Okay, since it's 1 mark, you save your time by just illustrating using the calculation. Now it says using the data in table 1.2, compare the mean volumes of carbon dioxide produced at 25 and at 35. Of course, what was change here, what was measured in between is at different times. The volume temperature used here was 25 and 35 at different time intervals. So we measured the mean volume of carbon dioxide produced as time was changing. Now what can we do? We are comparing. The question is 2 marks, so with 2 points we are good to go. So you don't need to take all the time comparing or wasting more time on a particular question. Look at the question and give 2 points. We see that on the overall, the mean volume of carbon dioxide produced increased, okay, both at 25 and at 35. So overall volume of carbon dioxide produced increased, okay, at both temperatures. All right, at both temperatures. That's 1 point. What does we see that on the overall at 35 degrees, the volume of CO2 produced was higher on the overall for 35 degrees. So we say degree C, the overall volume of CO2 produced was higher. Okay, that's 2 more. You can also say that for 25 degrees C, okay, for 25 degrees C, okay, no volume of CO2 was recorded at 5 and 10 minutes at the first, okay, at the first 5 and 10 minutes, okay, or for the first 5, for the first 10 minutes. Okay, these are some of the things we can use to compare. So you look at the table and bring out points that you see are worth comparing. Now we'll go to the next question. It says plot a line graph on the grid, okay, of mean volume of carbon dioxide produced against time using all the data in table 1.2. You will need to plot the data for each temperature as separate lines on your graph, including a suitable key. So we'll come to the table. We have at 35 and at 25. So in order to do this, we are plotting two line graphs on one graph book for 25 and 35 and using the same time. So of course, in plotting the graph, remember I told us that if you have been watching my video, I told us that so plotting the graph, there are certain things to take note of. First, You have to identify your independent variable, okay, and the independent variable is plotted on the horizontal axis, okay, on the horizontal axis. First, you also need to label your axis. You have to label your axis correctly with units. Okay, you have to label your axis properly, and you label them including the word unit. Axis with appropriate units, showing the units. Then you use a suitable scale. Use a suitable scale. Okay. You locate points and make your curve. Okay. From first point to the last point. Okay, so these are the things to take note of. Let's put that to work now. Following the table, we have the time is from 5 to 30. All right, so let's start with the time from 5 to 30. So we do this 1, 2, 3, 2, 3, 4, 5. So that's 5. Then 1, 2, 3, 4, 5. That's 10. Then we have 1, 2, 3, 4, 5. That's 15. Then we have 20. We have 25. We have 30. We have 35. Of course, labeling this axis with unit, the independent variable is time. The time here is in what? Time in minutes. So that is the axis. We label the axis as time in minutes. And on this side, we have volume, mean volume of CO2, mean volume of CO2. So that's the label here, mean volume of CO2 produced. Okay, what's the unit of the volume in cm cube? Mean volume per cm cube. So that's our label. We just this label alone, you have 1 mark. Then you go to your scale. I've done the scale for the temperature. The other one will be for the volume is from 0 to 3.2. So with this we can do this easily. Since we are going to 3.2, we can do 0.2, 0.4, 0.6, 0.8, 1.0, 1.2, 1.4, 1.6, 1.8, 2.0, like that, and then we get all the way to 3.2, 2.2, 2.4, 2.6, 2.8, 3.0, and 3.2. So that's to say that 5 units represent 0.2.
This is what we do in the masterclass, really. We try as much as possible to ensure that we're able to properly, right, teach learners how they can properly understand, interpret the question as much as possible. We dive deeper than just a past paper to ensure that at the end of the day, they're able to fully understand any question that is being administered to them.
2.2 cm cube on your graph. So that's what we have for our graph. And please always take note of your scale. Your scale must be such that it will cover at least 2/3 of the graph space provided. And with that, you are sure that you have used the scale alone will give you another 1 point. This graph is 5 marks, so you have 2. From labeling your axis, you have 1 mark. Okay, with the labeling of the axis using appropriate scale, you have 1 mark. Then we'll go to locating the points now. For 25 cm cube, we need a key. All right, let's say we use broken lines to represent the one that is 25. So this will be 25 cm cube graph, and then we use a continuous line to represent 35 cm cube graph. So that's your key right there. But doing that again, you have another point. Then let's look at the point. Now it says at 0.0 and 0. Okay, for 5 and 10 is 0, 0, and for 15 is 0.1. So let's do that. At 5 is 0, at 10 is 0, from 15 is 0.1. So in between 0.2 you have 1, 0.1 is just in between here. Okay, so that's that for that. Then for the other group you have 0.2, 0.5 and 1.0. 0.2, 0.2 here, 0.5 is in between 0.1, 2, 0.5 and 1.0. 1.0 is just here. So that's the graph for 25. So we just join the line like that. You can make it clear cut. Okay, so we have this, we have this, and we have this. So for this is at 0 and at 0. So we do this. Oh, sorry, we do this. Okay, so that's our first curve at 25. Then at 35, what do we have? We have 0 0.1, 0 0.8, 1.9. Let's go to 0 0.1. At 5, we have 0 0.1. Then we have 0 0.8. 1.9, 1.9, 2.7. So let's go to 1.9. 1.9 is somewhere here. Then 2.7. 2.7 is somewhere here. Then we have 3.2, 3.2. So for the last two units, it stays constant. Okay, so we have 3.2 and 3.2. So we have our graph like that. So we can just use your ruler or your free hand to make a clean sketch of your graph like that. Sorry, I'm using a drawing pen and it's not that accurate, but at least we have an idea of what we look like. So with this, our graph is set, and that's 5 marks already. All right, so that's 5 marks. So the important thing is using appropriate scale, using appropriate scale that will cover up at least 2/3 of your graph space. Let's see what are the questions that follow. It says estimate the time taken to produce 3.0 cm cube of carbon dioxide at 35. So all you do is come to your graph, and at 35, what do we have? Okay, so this is 1, 2, 3, 4, 5. So at 35, what do we have here? To produce 3.0, what do we have? So this is the time at 35 for 3.0, and that would be what? This is 5, 10, 15, 20. So that's around 25 degrees, around 22.5. Okay, that's around 22.5. Somewhere around here is around 22.5 or 23 minutes. Okay, so the time there will be what? 22, or let's say between 22 and 23 minutes, depending on what you have on your graph. So let me write what I can see, even though my curve is not that straight. So that's 23 minutes. And you don't need to repeat the minutes because the minute has already been indicated. So you have your 2 marks. Now it says carbon dioxide gas was bubbled through hydrogen carbonate indicator solution. The indicator was red before the gas was bubbled through. State the color change that would appear. Color change, of course, for increasing carbon dioxide, it will change from red, okay, to yellow, okay, yellow or orange, okay. Any of these two colors will be fine. So pick one. You can write yellow. Since they said color change that would occur, it's just write yellow or you write orange. You may not even need to show red to yellow or orange. So that's all with the first question. Now the second question is what we are going to go to now. Now, question 2 is another very interesting question to answer. And in question 2, it has to do with observing a specimen and perhaps drawing. Most times, let's look at what this one says. It says 52.1 shows the epidermal cell from a red onion. Now, let's go to it says draw a large diagram of the cell labeled X. Now, when they ask us to draw, there are certain things that our drawing must entail to get our questions. Number one, your drawing must be larger than the original one given. It should be larger than what you are given. And you ensure that you take note of these details. Number one, the outline. Okay, use a single continuous line. Okay, you draw with single continuous line, no shading at all. You don't shade. So you have an outline. Okay, outline. Then you have to pay attention to details, and you have to ensure that your diagram is to proportion. Okay, the details must be to proportion. So I summed it as S, O, P, and D. S here meaning the size, O there being the outline of your diagram, P there means your proportion. The diagram must be to proportion. All the parts of the details must be to proportion. So these are the things to take. These are the things to take note of when making your drawing. So let's put this to work. Now we see cell X. Of course, this is an epidermal cell from an onion leaf, so it's a plant cell. What are the things we can take note of? We can see these projections here. You can see this furrow here and here, and also this depression here. So these are some of the things we should take note of when we are drawing. Now in making your drawing, let's do a sketch of what we have. Remember, the size of the drawing must be bigger than what you have. And let me just make a free sketch. Okay, so in solving question 2, like I told us, we need to take note of these 4 principles. Number 1 is the size, the outline of your drawing, the proportion, and of course the details, essential details. Now I've made the screen into, I've copied the screen, moved it to another tab so we can have a clear picture of what we are drawing. So your diagram should be bigger, and use a clear continuous line. So that's what we have as our diagram. So ensure that you clean and avoid broken lines in between your drawings. So anywhere you notice a broken line, you can always erase neatly and make another sketch. So that's it. So we are trying to show the cell wall, as we can see. So we are showing the cell wall. So you have to just, it's an outline, it's not art. Okay, then we have, so that's what we have as our drawing. So at least we have all the details shown. Then here we have the nucleus or whatever it is. Just one single line is enough. One single line is enough. Okay, you don't have to make double line. Make sure you use a clear line. Okay, a single line for the nucleus. You don't shade. Okay, this is way bigger, so you have to make sure it's to proportion. Your diagram, everything must be to proportion. So right there, so that's what we have as our drawing. So if you notice any broken line, you can clean neatly and retrace. Make sure there are no broken lines and it's a single drawing. With this, we have our diagram ready. So we see the major details, the furrows, the depression here, here. Okay, so we have our depressions. Okay, so you make your drawing just simple enough, and these projections here and here, the position of the nucleus, and you have your cell membrane and so on. And that's all with the diagram. Let's now go on to the question. They said line PQ on fig 2.1 represents the length of cell X. Make the length of line PQ on the diagram, or measure the length of line PQ on the diagram and use the length. So all you just in measuring the length, all you just is place your ruler here and place your ruler at this edge and take the length of this line. You take the length, okay, of your drawing, and of course it's in millimeters. We know that in your ruler you have centimeter marks, and in between a centimeter mark you have the millimeter mark. So if this is 0 cm, this is 1 cm, so in between are the millimeter marks. Let's say this is 8 centimeters now, so in millimeters we have it as what, 80 millimeters. Let's assume that's what we have here. So they said if you are measuring with a ruler from your note, or sorry, from your script rather, and it says here, it says length of PQ. So you place your ruler and measure the length of PQ. Let's say it's 80 millimeters. That's how to take measurement. Now they said calculate the actual length of cell X using the formula and your own measurement. Okay, so calculate the actual length of cell X using the formula and your measurement. Okay, the formula and your measurement. So we have magnification already on the diagram here as 3 points. Okay, using the measurement, we have the magnification already here as times 340. So we know here that magnification is times 340. We have seen that the length here, let's assume with a ruler we got 80 cm, 80 mm rather, 80 millimeters. So using the formula, we need to know the actual length. So we know that magnification is image size over actual length. The image, of course, is, so image size from the original diagram given that we've measured is 80 mm. So we need the actual size. Actual size becomes image size over magnification. So we have 80 over times 340, and that's around 0.235 or thereabouts. Let's assume it's 0.235.
This is what we do in the masterclass, really. We try as much as possible to ensure that we're able to properly, right, teach learners how they can properly understand, interpret the question as much as possible. We dive deeper than just a past paper to ensure that at the end of the day, they're able to fully understand any question that is being administered to them.
4, 2. So they said we should keep our answers to 3 significant figure. So 3 significant figure will have your answer as 0.235. So all you just do is get your actual answer, and you have that in three significant figures. The unit is already there and you have your 3 marks. So going to the next question, this one says water moves in into and out of cells by osmosis. Plan an investigation to determine the effect of concentration of sodium chloride solution on osmosis in plants. Okay, so in this experiment you have to plan. It's a plan, it's not an action. You just give a guideline of what your plan should entail. And in planning an experiment, we need to take note of certain things. Independent variable, your dependent variable, the variables you keep same. Okay, variables kept the same, that's your variables that you keep the same throughout the experiment. You take note of your method and you take note of how you measure your result. Okay, the result and the instrument you are using, and you tell us a precaution or a safety precaution. Okay, precaution and safety. You also mention something about how you keep your results reliable, so you do reliability. So these points are the things that your plan should entail. Now we have seen that here we are looking at the effect of sodium chloride solution. So the sodium chloride solution becomes our independent variable. What are we checking? The rate of osmosis in a plant tissue. So we can decide to use a potato cube, all right, from a potato tuber plant. Now how do we do this? So let's start with our investigation. First you have to talk about how you get your independent variable. So you say for independent variable, you said make, at least you can do 2 or 3 concentrations. Make 2 or 3. Okay, make 2 concentrations, of sodium chloride solution. You can say you do 0.4 mole per dm cube and 1.0 mole per dm cube, and distilled water. So you can also add distilled water and distilled water. Okay, in three test tubes, in three test tubes. So these are planned in 3 test tubes. Okay, with labels. That's how you make your, okay, with labels of same volume. Okay, of same volume. So you have the same volume of 3 different concentrations of sodium chloride solution. So you have distilled water 1.0 and 0.4. So that will make 3 different concentrations of sodium chloride. So that will be 3, make 3 concentrations. So distilled water you can include and you can decide not to include, but I think it's better we include this. So that's our independent variable. What are we going to measure? We are going to first look at our methods and what we keep the same. So method, we are going to cut, okay, 3 pieces of potato cube, okay, of same length and from same plant, same length and from same plant of same mass too. So you are going to have it as same length or same mass. Okay, same mass. All right, then you, let's also put how we will cut. So we'll cut, okay, you might include that you cut on a tie or something. Once you have done this, Next thing is for how you get your dependent variable measured. So we say we put in terms of method. Now once you've cut, so you say in each test tube, okay, put each potato cube and cover the test tube or cylinder, cover the test tube with a tight lid. Why are you covering with a tight lid? To prevent evaporation of water of water. What else are we going to do in our method before we now take our result? Okay, leave the setup for, let's say, 1 hour. Let's use 1 hour. For 1 hour, leave the setup for an hour. Leave the setup for 1 hour. Okay, then remove each piece, dab each piece with dry a dry cotton, or napkin. Okay, with a dry cotton, you could use a napkin and measure, okay, the change. You can now decide to measure the change in mass, or you measure the change in length. Anyone, if you want to go with measuring the length of the tube, or you want to go with measuring the mass of the potato cube rather, then you can measure a change. Take note of this change in what? In mass, or change in length of each piece. So that's how we get your results. When you measure, what instrument I use to measuring the change in mass? Using a what? A balance, a weighing balance. So stating the instrument will give you at least a mark, because these are the things that they want to see. So you get your balance. Then what are the things we have talked about? Our result and the instrument you use to measure it. We've talked about the method. We've said we've looked at the things we kept the same, and we've talked about our dependent variable. What precaution or safety are we going to do? And reliability. So for precaution of safety, you say ensure, okay, to cut. You ensure to cut on a flat surface. While you are cutting the piece on a flat surface away from the body, okay, so you don't injure yourself. And that's your precaution. Then reliability, you can go on to say you the experiment repeat the experiment for each concentration 3 times. Okay, you can say 3 times. Okay, and take average, excluding anomalous result. I think with this we have more than 6 marks. So because it's a 6 mark question, with this you have more than 6 marks. So you have the concentrations, independent variable sorted. So your independent variable sorted, and how you get it, you have 2 marks there. How you make your potato of same length and same mass, you have 1 mark. the test tube recovering with a lid. You have 1 mark for that to prevent evaporation. You also talk about the duration. You leave it. These are some of the variables you kept the same. The duration of the experiment, how you ensure that your result is reliable. You dab with a dry cotton, and what you are measuring, change in mass, of course, the instrument, then precaution and reliability. So with this you have more than 6 marks. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 already. So any of this would be enough for you to get your maximum mark. So I think that's all with solving this paper. So ensure that you always take note of these details. You can watch this video again, and you see how to do this. With practice you can always get maximum marks. Students get as much as all the points present in this paper.
This is what we do in the masterclass, really. We try as much as possible to ensure that we're able to properly, right, teach learners how they can properly understand, interpret the question as much as possible. We dive deeper than just a past paper to ensure that at the end of the day, they're able to fully understand any question that is being administered to them.